Welcome everyone to our May Lunchtime lecture. I'm Patricia Shampton, Texas Ex's Director of Alumni Relations, and we're so glad you could join us today. But before we begin, I want to point out that we're just a few days away from the big commencement ceremony for the class of 2022, which will be held this year at DRK Texas Memorial Stadium. If you know a Longhorn who is about to graduate, we have the best gift around. Give them a Texas Ex's Life membership. Now through May 22nd, new grads can save up to 40% on the Texas Ex's Life membership. Just visit texasexes.org/graduation to learn more. Okay, so now on to our program for today. Our guest is Dr. Lauren Ehrlich, and her lecture today is T for Terrific, How T Cells Protect You Throughout Your Life. Dr. Ehrlich is an Associate Professor of Molecular Biosciences and Oncology at the University of Texas. She did her undergrad at Yale University and got her PhD in immunology from Stanford University Medical School. In addition to her teaching work at UT, she is a member of the LaMontagne Center for Infectious Diseases and the Livestrong Cancer Institutes at the Dell Medical School. Her lab focuses on identifying the cellular and molecular mechanisms that govern differences in diverse and non-autoreactive and non-malignant T cells. Current research in the lab also includes investigation of immune signatures of disease severity in COVID-19 patients. Please welcome Dr. Ehrlich.
Thank you so much. Can you hear me okay, Patricia?
Yes, you are perfect.
Perfect. Thank you and welcome everybody. First, thank you so much to Roxanne and Patricia, the Texas Exes, for inviting me to come and talk to you today. As Patricia told you, I'm Lauren Ehrlich. I'm an associate professor in the Department of Molecular Biosciences and also in the Department of Oncology at Dell Medical School at UT Austin. And really, I'm an immunologist. So I want to tell you a little bit about why T cells fascinate me and their various roles in protecting you throughout our life. And then really the thing that I get very excited about is how T cells know how to attack all of these different pathogens that we encounter throughout our lives without attacking our own bodies and causing autoimmunity. So that's sort of a major focus in the lab. We're going to talk about a little bit today and how that changes over our life. So you can kind of see here, I love watching T cells, and what you're seeing in these movies on the bottom left and bottom right are developing T cells as they migrate around. So the developing T cells are red over here, and they're migrating around in this thymus. And that's where they're developing. On the right, you can see them interacting. They're the purple T cells over here on the right, and they're interacting with these yellow dendritic cells. And so we'll talk a little bit about what are these T cells learning in the thymus and why is that important for a healthy immune response throughout life. All right, so I thought I'd just back up because I don't really know how much in terms of who's out there in the audience, how much immunology background you have. So I just want to start by saying our immune system is essential, really critical to protect us against the barrage of pathogens that we encounter throughout our lives. And that might sound like a really big over-exaggeration, but it isn't. And the way we know that it isn't an exaggeration is that our cases like this. So you guys are probably all familiar with David, who was a boy who lived in Houston. This is back in the 1970s, where David was born without an adaptive immune system. And we know that kids who are born without an adaptive immune system, so these are our T cells and our B cells, will succumb to infectious disease unless at this point they receive a bone marrow transplant, very early in life that helps to provide an immune system that can protect them. And so there was another really interesting thing about cases like this. So these are kids with severe combined immunodeficiency where they don't have T cells and B cells. So not only are these kids susceptible to infections, but they're also susceptible to cancer. And that's because our immune system it's really got this amazing job. It not only fights off all of the different viruses, bacteria that we're encountering throughout our lives, but it also fights off newly formed tumors. And eventually, David, as a young boy, ended up getting a bone marrow transplant, but the bone marrow was EBV infected, and EBV is a virus that actually infects some of our immune cells and causes leukemia. And he ended up dying of that leukemia because he didn't have a healthy immune system to fight off the virus that caused a cancer. So there's this constant, as you heard, my lab is interested in infectious disease and how T cells respond to that. We're also interested in cancer, and that's because the immune system plays an essential role in fighting both infectious disease and cancer. Okay, so let's just walk through what are key functions of a productive immune response. So I think, we all think about an immune response. You get sick, it fights off an infection, you're healthy. But what is really going on in an immune response? It's complicated. So the first thing that happens is, let's say that you get infected with some virus. I mean, I just happen to pick a random one, right? I happen to pick SARS-CoV-2 right now. That comes in and maybe we have a lung infection. So the very first thing that our immune system has to do is to recognize that there's a problem, right? And so resident in tissues throughout our body, we have cells of the innate immune system. So here's a macrophage. And the first question that macrophage has to ask is, look, I'm kind of seeing something. Is this friend or foe? Do we really have a problem here? Do we need to mount an immune response? And once the macrophage makes that decision and it decides that there's a problem, it's going to initiate the immune attack. And it's going to do that by calling in other cell types and activating other cell types in the immune system that are going to help to eliminate the pathogen. So in come a whole lot of other immune cells. So our T cells, our B cells, natural killer cells, and neutrophils. So these are components of different. The immune system is a layered process where you start off with sort of this innate response. Macrophages are part of it that recognize that there's a problem. And you bring in other innate cells that can sort of, at low level, keep the virus or bacteria from getting really out of control before you recruit in your adaptive immune system that's comprised of your T cells and B cells that make this very specific response to a particular pathogen like SARS-CoV-2. But you're going to basically, in initiating an immune response, once you recognize that there is something foreign that we need to mount a response to, bring in all of these cell types and allow them to get activated to carry out their effector function. In other words, to help eliminate the pathogen. So once that happens, that process is really important for protecting us from pathogens. But at the same time, and this is something we don't really tend to think about too much, right? It's not only important that we mount an adaptive immune response and allow the pathogen to be cleared, but then we have to dampen down the inflammation caused by the immune response itself. And that's something that's become, I guess, that's more in our collective conscience over the last couple of years, because we know that patients who are responding to SARS-CoV-2, when they have too much of an immune response, sometimes because there's too much of a viral load, too much virus around, that really potent immune response is actually damaging to our own lung tissue. So controlling the immune response and dampening it back down after clearing a pathogen is also a really key feature of regulating a good immune response. And then finally, once you've cleared the pathogen, you don't want to start over from baseline again, right? We want to make sure that once we've encountered a pathogen, once we have the ability to remember it, so that if we re-encounter the same pathogen, like if we get reinfected with flu or reinfected with SARS-CoV-2 and COVID, that we have the immune system is wiser now, and it's got these B cells that are pumping out antibodies and T cells that can carry out other functions to clear the virus, and they have remembered that specific virus. So we've got these wiser T cells and B cells that are our memory cells that can help to more rapidly mount a response and clear an infection before you even really know that you're sick and have re-encountered that pathogen a second time. So those are sort of key features of a productive immune response. And we're going to really focus in today on the role of T cells in this adaptive immune response because, as my title says, T for terrific. So we're going to talk about how T cells know how to recognize foreign pathogens and fight them off, but somehow at the same time manage not to attack the cells and tissues, the diverse cells and tissues throughout our own bodies. The immune response has this remarkable challenge, right? You know, we're just talking about SARS-CoV-2, but there are all sorts of pathogens that your immune response needs to protect us against. And they're really diverse in a variety of ways. So one of the ways in which these pathogens are diverse is that they're located in different regions in our body, right? So our body, all the tissues in our body are made up of a lot of cells. And those cells, are connected through various junctions, but there's also a lot of extracellular space. So pathogens can exist in extracellular space, and we can have bacteria there. Actually, viruses can have an extracellular part of their life cycle. There's parasites and fungi. And so our immune system needs to be able to find and recognize pathogens in extracellular space, like Streptococcus pneumonia that causes pneumonia. And those pathogens can exist on really different scales. So some of them are really tiny, like micron size, 10 to the minus 6 meters, so micron size for bacteria, or on the millimeter scale for fungi. And then in addition, we have intracellular pathogens, like again, some bacteria live inside of our cells. Some of these pathogens can actually get in and live inside of cells, and so can parasites. So for example, leprosy is a case of an intracellular infection, and our immune system needs to be able to find those pathogens that are actually within our own cells and somehow attack and get rid of the infected cells along with the pathogen. And then there's, again, just very different size scales of these pathogens. So we were talking about micron and millimeter scale for bacteria and fungi. Well, let's look at viruses. Now we're talking about nanometer scale infections. So tiny little viral particles that somehow our immune system is seeing those too and helping to clear them. And then on the complete opposite side, we have, parasites like worms that can infect us, and they're on centimeter size scale or bigger. So this is a lot of diversity in sort of the types of pathogens the immune system can recognize, the size of pathogens, and the location within our cells and bodies that the immune system needs to be able to survey and protect us against. So T cells are really key to recognizing not only all these diverse pathogens, but also tumors. And they are our key regulators of the adaptive immune response. This is the really specific immune response that's going to recognize this year's influenza virus versus next year's strain and be able to discriminate between them. And then really remember what we've mounted an immune response again and respond quickly in the future. These are key cells. So what are some of the diverse roles that T cells, one subtype of this, one subset of the immune response, what do they do? So first of all, there's two major flavors of T cells. There are helper T cells, also known as CD4 T cells, because of a molecule called CD4 that they express. And these helper T cells have a whole variety of different functions. So just to name a couple of them, during the course of an infection, T cells will actually interact with the other major subset of adaptive immune cells, our B cells. And they'll recognize a B cell that's going to make a good antibody response. And they'll provide signals to that B cell that make the B cell secrete antibodies and form a really good, highly protective antibody response. So even when we talk about, like, let's say, you've gotten your SARS-CoV-2 vaccine, you know that you're going to have a lot of antibodies that are going to protect you against severe disease or infection. And we talk about how antibodies are really key. Well, B cells are making antibodies, but your B cells aren't going to make good antibodies without help from your helper T cells. So again, the helper T cells are really key to mounting this adaptive immune response and clearing extracellular pathogens. So antibodies are surveying extracellular space. But your CD4 helper T cells can also help with intracellular infections. So they can recognize if you have a macrophage that gets infected with like a mycobacterial infection. And so the bacteria like to hide out. Macrophages are great at gobbling up things in the extracellular space and looking for infections, but bacteria have taken advantage of that and they can actually live inside vesicles at macrophages. Well, T cells can recognize these infected macrophages and help, again, they provide signals to activate the macrophages to clear the intracellular infections. And then the other major flavor of T cells are cytotoxic or killer T cells, also known as CD8 T cells because they express a different molecule, CD8, on their surface. These cytotoxic killer T cells can actually directly kill infected cells. So if you have cells with a viral infection, your killer T cell can recognize and eliminate that cell. They can also do this for for cancer cells. So newly arising cancers can be recognized by our killer T cells, and they can actually literally cause those cells to die while leaving the nearby healthy cells intact. So it's a pretty remarkable discriminatory capacity, right? To see an infected cell and leave your neighboring cells that look very similar to that infected cell alone. And then the final subset of T cells that are important to mention for what I'm going to tell you about are regulatory T cells, or we call them Treg cells. So this is just a different category of cells that also express CD4. And they have a job that's completely different from our helper, our T cells, and our killer T cells. So you guys, if you see anything about, if you read about T cells or you hear about T cells in the popular press, you're normally going to be talking about these helper and killer T cell subsets. But regulatory T cells are also super important. And this has to do with what we were talking about earlier, that your immune system is really protective against pathogens and cancer, but it also can be really damaging and cause a lot of inflammation. And so we need to make sure that the immune system is kept in check and does not cause overt inflammation or overt autoimmunity. And the job of our regulatory T cells is to make sure that we're going to suppress autoreactive T cell responses that could otherwise cause autoimmunity. Okay, so how is it possible that this one cell type can recognize, so many diverse pathogens, newly arising tumors, and do this with incredible specificity? So it can recognize, let's say the Delta variant of SARS-CoV-2, but perhaps not the Omicron variant. There are some T cells that can recognize both. Okay, so how is that possible? So I'm going to step back for a minute. Well, let me first tell you how it's possible, and I'll tell you why this is such a mind-boggling number. All right, so here's your T cell, and we're going to delve in a little bit more to the molecular mechanisms of how T cells can recognize specific pathogens. So here's your T cell, and every T cell expresses on its surface a receptor that's made of two different proteins, an alpha chain and a beta chain of the T cell receptor, or TCR. So yeah, the very non-creatively named T cell receptor is what confers specificity on a T cell. It's what lets the T cell recognize a particular pathogen infection. And this T cell receptor, what it does is if we have an infected cell down here, let's say we have a cell with a viral infection, flu, what your cells do all the time in your body is they chop up proteins that are inside the cell. Some of those proteins, if the cell is infected, are going to come from the pathogen itself. And then they display those proteins as little pieces of the protein on the surface of the cell within these molecules that we're just going to nickname for the moment MHC molecules. And I'll show you what those look like in a minute. So all the time on the surface of your cell, you've got these cells, you have these MHC molecules, and they're displaying little pieces of what's inside of them. And that they're displaying that so that your T cell can come down and query this peptide, this piece of the virus or piece of a bacteria or piece of your own protein. So it can look at those proteins and say, hey, is that something that I recognize? And if I recognize it, that T cell is going to get activated and mount an immune response. And what do I mean by recognizing it? So let's look at a little bit in a little bit more detail. So this is what the crystal structure of a T cell receptor looks like. So again, you can see these two chains in blue and in turquoise, just like over here. This is the alpha chain and the beta chain of the T cell receptor. And down here, what we're looking at is this MHC molecule in brown with the yellow peptide. And you know what the MHC molecules look like? They look like hot dog buns on a stalk. So basically, it's like you've got this stalk that's sticking off of the surface, and there's lots of these all over the surface of our cells. And then you have these two alpha helices. You see these little spirals that are here that look like the sides of our hot dog bun. And stuck down right in the middle of the hot dog is the peptide. The peptide is a piece, it's amino acids, a short string of amino acids, a piece of the proteins from inside the cell. So this is how all of our cells are displaying pieces of pathogens or tumor antigens, pieces of tumor that doesn't look like us, to our T cell receptor. And the T cell receptor is reaching down with these finger-like projections that are shown in blue, orange, yellow, pink, and it's seeing if those bind to this particular peptide, if they sort of stick tightly. If so, that T cell gets activated. Okay, so that's the basis of the specificity for a T cell to see a particular antigen. So for example, this peptide might look different if it's this year's influenza versus next year's influenza. The proteins change in flu from one year to the next, and so this peptide is different. So the T cell receptor contact with that peptide will change. Maybe it's not strong enough to see next year's flu, but it was strong enough to see this year's flu. And that's the T cell that got activated to this year's flu. All right, so that's sort of the basis of recognition. But the mind-boggling number is this. So, all right, I told you there's this specific protein and it reaches down these finger-like projections, and that's how you're going to discriminate the different, pathogens. But there's a lot of pathogens out there, right? Viruses, bacteria, fungi, proteins that are mutated in cancer cells. So how many different T cell receptors do you think you would need to see all of these different possible antigens and be able to respond to anything that's out there, newly emerging viruses like SARS-CoV-2? A lot, right? Like a ton. Well, it's estimated that we can express 10 to the 18th different T cell receptor specificities. So our T cells can make 10 to the 18th different T cell receptors. Now, that's just like a mind-boggling number that I can't even wrap my head around, right? But it's plenty to recognize all the diverse, more than enough to recognize all the diverse peptides that are out there. So that's great that that happens. But here's the conundrum. I don't know, some of you probably know this, but you would think, because we have so many diverse cell types, we've got all the cells that make up our retina, our neurons, all the support cells in the brain, our intestinal cells, right? Like tons of different cell types. And those cells need to express different combinations of genes in order to be the cell type that they are, to be a neuron versus a, epithelial cell in your skin. So you would think we have just this huge genome with tons of different genes. And so it wouldn't be any problem to encode 10 to the 18th different T cell receptors. But it is a problem. And that's because we actually only have about 20,000 genes in our genome, which is a remarkably small number, much smaller here than the 10 to the 18th here. 20,000 genes that somehow are encoding all of the proteins that make all the different cell types in our body. So how on earth can you take 20,000 genes that are encoded in your DNA and make 10 to the 18th different T cell receptors? So this is one of the cool things. Like, if you've taken intro biology, you'll learn that you have, in your DNA, you've got genes. The genes are turned into messenger RNA. That messenger RNA is translated into a protein, and that protein does the business end of whatever the protein needs to do to make the cell function properly. T cell receptors are encoded differently. So instead of having one T cell receptor alpha chain gene and one T cell receptor beta chain gene, because remember the TCR is made of 2 protein chains. Instead, we have all of these diverse gene segments in our genome that encode the T cell receptor alpha chain and the T cell receptor beta chain. And when T cells develop in our thymus, I told you we'd get back to the thymus, as these T cells are developing and becoming mature T cells that are going to be able to protect us, these gene segments are rearranged at the DNA level. So our DNA is being spliced and broken up as T cells are developing. And these segments of the T cell receptor are being literally brought together at the DNA level, both for the alpha chain and for the beta chain. And there's multiple choices of which gene segments to use. And that combination of using a bunch of different gene segments allows us to encode these diverse T cell receptor proteins that have very different finger-like projections that are touching the peptide MHC complex, the hot dog peptide bun, to recognize a specific pathogen. So that's how we get so many different T cell receptors. But there's something really inherently dangerous about this process. The DNA is being spliced up. These T cell receptor gene segments are being joined randomly. And that means that by chance, some of them will do the job we want. They'll recognize SARS-CoV-2 spike protein, or they'll recognize, hemagglutinin from influenza. But some of them, by chance, some of the newly generated T cell receptors will happen to bind peptides that come from our own proteins. So if you remember back here, these peptides that are being displayed on the hot dog bun on the surface on all of our cells, they can come from a pathogen, but they can come from our own proteins. And a matter of fact, most of the time when we're not sick, what our cells are doing are displaying our own proteins to T cells. Okay, so if you can randomly generate T cell receptors that recognize those proteins that make up our neurons, that make up our retina, then all of a sudden we've got a problem, right? Because those T cells then have autoimmune potential and they could potentially cause autoimmune disorders. So that's something that really fascinates me. How does a T cell know? How do we educate our T cells to make sure that they're responding only to foreign pathogens, only to cancer, and not causing autoimmunity? So T cells develop and are educated in the thymus. So the thymus is an organ that you may have eaten, possibly, but that you probably otherwise haven't paid a lot of attention to. It sits right over our heart. And basically cells from the bone marrow, this is where all of the cells of our immune system develop, where the precursors to all of the immune systems are. Those cells are going to actually leave the bone marrow. They're going to traffic to the thymus and seed it. And within the thymus, these cells, we're going to look through this process in a minute, are going to decide to become T cells. And they're going to go through that process of rearranging their T cell receptor genes, and they're going to get educated to make sure we can have a diverse repertoire of T cells that can see all the pathogens without killing our own cells. So here's what this looks like. This is an immunofluorescence image of a thymus, and you can kind of already see that there's two, well, along with my blob of a schematized cell here, but you can see that there's two major regions. There's this outside region in red that we call the thymic cortex, and there's this inner region in green that we call the thymic medulla. So two major compartments of the thymus. And what happens is those progenitors that leave the bone marrow seed the thymus through blood vessels that are near the junction of the cortex and the medulla. And as they start to differentiate and decide to become T cells, so they haven't made up their mind yet. When they first hit the thymus, they could become B cells, they could become T cells, they could become dendritic cells, another cell type in the immune system we haven't really talked about. But once they hit this thymus microenvironment, most of them decide, I'm in the thymus, I'm going to become a T cell. And when they make that decision, they start to rearrange those T cell receptor beta chain gene segments so they can encode a T cell receptor. So the first thing that happens, the first major checkpoint in T cell development is that the T cells say, hey, did I make a productive beta chain rearrangement? You can imagine if you're splicing up DNA and you're throwing together gene segments, sometimes you just don't encode a good protein at all. And we don't want those T cells out in our circulation, gumming up, taking up space for productive T cells. So we want to get rid of those. So that's the first checkpoint. If the cell has made a productive beta chain rearrangement, it gets a signal to proliferate. That's my little circle here. And you expand out a lot of progenitors that have these functional beta rearrangements. The cell then goes on and starts to differentiate to this double positive fate. Don't worry about that. But it rearranges T cell receptor alpha chain genes. And so that means that, since all of these cells are making a good beta chain because they passed beta selection, once they've made an alpha chain, the TCR can be expressed on the cell surface as this alpha beta heterodimer. And now this developing thymocyte can start to look around and say, hey, do I recognize anything through my T cell receptor? This is the next really critical checkpoint during T cell development, where the T cells say, just ask if they made a functional T cell receptor. And by functional, we mean, does it have any capacity to recognize those hot dog bun MHC molecules? Because if so, it could potentially recognize a pathogen. And so if so, the cell proceeds and it differentiates. If not, it dies. And actually, there's a lot of cell death through these processes. So like 95% of the cells of the developing thymocytes that go through this, thymocytes are just developing T cells. 95% of the developing T cells actually die along the way. So they don't make it through these functional checkpoints. If they do make it through, then they're going to move into the medulla, this middle region of the thymus, and they're going to start to differentiate to become the precursors of our helper T cells or our killer T cells, so the CD4s or CD8s. But they stay in the medulla for a little bit, and we're going to zoom in and look at that. And that's where we make sure that these cells are not going to recognize our own tissue and our own cells and cause autoimmunity. So this process of the thymocytes, the developing T cells coming into the thymus, migrating around, going through these complicated processes of differentiation and selection to ensure that we've got a good T cell response are really what fascinate me and drive a lot of my lab's research. And as you can kind of already see from the immunofluorescence data, the thymus isn't just a bag of developing T cells. There are a lot of other cell types in the thymus that these developing T cells are interacting with that are providing signals and cues to help the T cells during their education. So these are our teachers in the thymus. Okay, so let's zoom in for a minute to the thymic medulla. That was the central region of the thymus where tolerance occurs. So this is where we make sure that our T cells are not autoreactive. So once we have our helper and our killer T cell progenitors in the medulla, they're going to actually look around, and what they see are these, again, hot dog bun peptide MHC complexes that are expressed by different cell types in the medulla. So the teachers of the thymus. So we have medullary, that's the M, medullary thymic epithelial cells, so epithelial cells in the medulla. And these are really cool cells that I'm going to tell you about. And then we have another major important subset for presenting our own proteins to developing T cells, and these are dendritic cells. So the idea is that if by chance these T cells have expressed an autoreactive T cell receptor through that process of gene rearrangement, they're going to run around and they're going to look at the peptide MHC complexes presented by these dendritic cells and MTECs. And those peptides are going to come from our own proteins. They're going to come from proteins made by a medullary thymic epithelial cell or made by a dendritic cell. If those T cells recognize through their T cell receptor those self-antigens, that tells the cell, hold on, I'm seeing self-protein in the thymus, I'm autoreactive, I need to die. So that's going to provide this strong signal through the T cell receptor that's going to cause cell death of these autoreactive T cells. On the other hand, we know that this process isn't perfect, and I'm going to tell you why it's not perfect in just a second. Well, you guys already know it's not perfect, because if it were perfect, we wouldn't have autoimmune disorders, right? And you know, probably everyone here knows someone with some form of autoimmune or autoinflammatory disorder. And so that's when those processes of tolerance go awry. So it's really a fine balance in our immune system of making sure T cells can see all the pathogens and not cause an autoimmune or autoinflammatory problem. On the flip side, remember those special Tregs. So the regulatory T cells, they also get selected in the thymus. So some of these cells, and this is a very interesting decision we still don't fully understand, some of these helper T cells, when they see a self-antigen, instead of dying, they become a regulatory T cell. And that's really important, because those regulatory T cells can then actively suppress any autoimmune T cells that escape this process of tolerance in the thymus. So if you so why would you ever escape the process of tolerance? Why haven't we set this up to be perfect, right? So part of the problem is every cell in your body, right, expresses a subset of genes, and that subset of genes makes the cell what it is. So again, can make your cell, you know, your neuron, or can make a cell the taste bud on your tongue, or the epithelial cell in your skin. And it's different combinations of genes that are expressed that make the right proteins to basically have the building blocks for the specific cell type. So if you think about a medullary thymic epithelial cell, you know, once your T cells develop and they get out to the body, into the rest of your body, where they're going to scan around, like we talked about, and look for infections and cause an immune response. You don't want your T cells to be tolerant only to the particular set of proteins that a medullary thymic epithelial cell expresses to be a medullary thymic epithelial cell, or a dendritic cell expresses to be a dendritic cell. You want your T cells to be tolerant to your retina, to be tolerant to your neurons, to be tolerant to proteins expressed in your joints so that you don't get rheumatoid arthritis, right? Okay, so how is it possible that our T cells are educated to all of these diverse proteins that really should only be turned on by cells in differentiated tissue elsewhere in the body, in differentiated organs? And this is this amazing feature of our medullary thymic epithelial cells. So collectively, they express over 90% of the genes in our body. And that's pretty remarkable. So they express genes like insulin that should otherwise be expressed in the beta islet cells in our pancreas, or IRBP that is expressed in retinal cells. And those genes don't really have any functional business being turned on in the MTECs. They're turned on at low level to make sure that this looks like a little library in the medulla, a display library of all of the proteins in our body the T cells are going to encounter when they get out into the rest of the body, so that we induce tolerance. But if you tried to express 90% of the genes in one cell, that cell would really undergo a lot of stress and die. That would not be good for the cell. It can't handle it, too much protein load. So instead, any one of these antigens that we call tissue-restricted antigens, these diverse proteins, are expressed by only 1 to 3% of these medullary thymic epithelial cells. So we kind of have this sparse mosaic of self-protein display throughout the medulla, and the thymocytes have to really get in quickly and rapidly scan all of these different peptide MHCs to find out if they're autoreactive or not, to find the self-antigen that might be really rare that's expressed by an MTEC. Dendritic cells also express self-antigens, and they have some other cool properties. So they can pick up antigens from blood and present them to thymocytes to induce tolerance. They've actually been shown to be able to pick up proteins from like extracellular space in different tissues, pick them up, traffic those proteins to the thymus, self-protein, and show them to developing T cells to induce tolerance. And then they can also help to pick up antigens, and we still don't know how this works entirely, that are expressed by these medullary thymic epithelial cells and display those antigens more broadly. So it's not just 1 to 3% of the cells, but a little bit broader display of given self-antigens. And that's really important. So we know that without proper central tolerance, we get autoimmunity. And there's a whole list of different, possible autoimmune disorders, right? Lupus, RA, and other autoimmune disorders, multiple sclerosis. So we know that if tolerance is impaired in the thymus, those autoimmune disorders can develop. And so my lab is really interested in, okay, just to give you a big overview of what we're interested in, how do T cells develop and undergo selection throughout the lifespan? So how do they retain reactivity to the diverse pathogens while avoiding autoimmunity? And how do these processes governing T cell education change to meet immune needs throughout life? And hopefully I'll have time to touch on that a little bit. And what you can see here, and I'll show you another movie of this in a minute, are T cells migrating in the thymus, and this central region where you can really see a lot of them here is the medulla. So I should tell you that as T cells develop, they had about 4 to 5 days to find all those self-antigens before they mature and even go out to the periphery. So they really have to get in there efficiently and scan antigens efficiently to undergo full central tolerance. Things I'm not going to tell you about, we're really interested in this pediatric malignancy, T-cell acute lymphoblastic leukemia, which is a common pediatric malignancy, and it's a cancer, it's a leukemia of developing T cells. So we're very interested in what are the cells and signals in the tumor microenvironment. So as this leukemia starts in the thymus, what cells are those leukemia cells integrating from cells in the environment that help to promote tumor progression? Because if we can understand those signals, we can block them and cut off leukemia progression. So that's another aspect of what my lab is working on. And then, though I'm not really going to end up touching on this, we're doing a lot of work looking at immune correlates of disease severity in COVID-19 patients. And again, really looking across the lifespan, because we know that severity has been associated with advanced age, and that kids in general have been more protected. And so we know there's a lot of changes in the immune response throughout age. And so we're interested in determining what immune correlates are changing across the age span, and trying to understand whether or not some of the differences at baseline in people of different ages are helping to contribute to this disparity in severity in different ages. All right, so I keep talking about different ages of your immune response. So here are some differences. So what we're looking at here is sort of a schematic of what happens from the time we're born as we age. And if you look at thymic T cell output, what's interesting is that, when we're first born, there's sort of this moderate, well, before we're born, there's, really not a thymus. The thymus has to develop, and then eventually it has to get those progenitors that seed it and start to make T cells. And so eventually we start to output T cells. And at sort of really a very young age, we have maximal T cell output from the thymus. And then the thymus starts to shrink, it starts to involute. And as we age, you get fewer and fewer T cells that are output from the thymus. But along with that change in thymic T cell output, there are other real age-associated changes in T cell responses. And let me just back up for a minute and say, if you think about it, okay, when you're first born, you're going to encounter all these pathogens you've never seen before, things that, you know, you and I fought off when we were in our childhood or infancy, right, or got vaccinated against. And so we've mounted immune responses, and we kind of haven't thought about those things since then. But when you're born, you haven't encountered any of them, and you've got to mount new responses to all of these different pathogens. And so it's really important that your immune system mounts very rapid responses to clear pathogens and protect newborns against many, many different pathogens they encounter when they're first born. And there's some special features that we're starting to recognize of immune responses in the neonatal period. So the conventional T cells, meaning not the Tregs, the conventional T cells that mount our immune responses that are educated in the newborn thymus, interestingly mount faster responses to pathogens. So there's more naive cells produced. They make a lot of these really fast innate-like responses, but they don't do a good job of making memory responses. Very interestingly, regulatory T cells that are educated in the newborn thymus are more suppressive. They seem more autoreactive, but not in a bad way, in the sense that they're better at recognizing autoantigens and suppressing autoreactive T cell responses. And there's some evidence from mouse models that these neonatal Tregs are critical for suppressing autoimmunity throughout life. So that's really interesting, and we don't understand what's different in the neonatal thymus that's selecting these super protective Tregs. But you can imagine that if we could figure that out, which is something we're looking into, then you could actually use those super suppressive Tregs to dominantly suppress autoimmune disorders. And then as we age, and the thymus involutes and there's less T cell output, so the number of naive T cells goes down. Naive T cells are just T cells that are circulating that haven't yet seen a pathogen but have the potential to respond. So our number of naive T cells goes down, but you know, our immune system has gotten wiser. We've seen a lot of pathogens throughout our life, and so we have more memory T cells. So our ratio of memory to naive T cells goes down. But that does mean that if we're trying to mount immune responses to new vaccines or new pathogens, we don't have as much of a starting pool of T cells to make a new immune response with. And so this sort of leads to impaired T cell responses with age to vaccines, pathogens, and newly arising cancer. And then this has led us to this question that I'm going to briefly touch on, which is, are T cells generated in this shrinking thymus? Is it just that there's fewer of them, or are they different? And in particular, we were curious about whether or not T cells that develop in this involuting thymus are sufficiently tolerized to self-antigens to prevent autoimmunity. And part of the reason that we're interested in that is that really at middle age, although the thymus is not massively involuted, that's where you really see a peak in new onset autoimmunity for a lot of autoimmune disorders as this thymus is starting to shrink. So we wanted to know if the involuting middle-aged thymus was capable of inducing self-tolerance. So again, thymic involution diminishes naive T cell output with age. So if you look at the functional thymus tissue, this is mouse thymus tissue, you can see in a young adult that there's kind of this large organ, and as they age, there are a lot of changes that are going on, but part of it you can just already see kind of this shrinking cortical region and an overall smaller organ. Along with that, if you just look at the cell numbers over the first 12 months of age, this is again a mouse, so this is about a middle-aged mouse, they live about 2 years of age, you can see that the total number of cells in that thymus is really declining, and there's a rapid fall-off. So even between 1 and 3 months of age, both of which would be considered a pretty robust adult, there's a dramatic fall-off in thymus cellularity and export. So this is work from Pam Fink's lab that showed quite a while ago that if you look at T cells exported from the thymus, and this is now in weeks of age, you can see that there's this real peak very early on within the first month of age, and then there's this precipitous decline in T cell output. So by the time that they're a middle-aged mouse, there's much lower T cell output. It's not gone. Similarly, in people, you can see that in a newborn, so here's a 5-day-old thymus, you have this really robust thymus tissue. You can see the cortical region in the dark purple and the medullary region in the pink. And in people, there's lots of lobules of the thymus with cortical and medullary regions. But even by the ripe old age of 28, you can see that there's not nearly as much thymus tissue, and instead your thymus starts to be replaced by adipose tissue, by fat. And by the time that you're out in close to your 8th decade of life, there's a little bit of functional thymus tissue left, which you can kind of see in the pink and purple, but very, very little. So it's largely been replaced by adipose tissue. And similarly, if you do a similar study here, this was work from Danny Duick and Greg Sapolsky and Lynch that showed that if you measure T cells output from the thymus with age, and so now this is looking at a log scale, right? So 10-fold changes here, and you look over age, that by the 8th decade of life, you're really putting out very, very few T cells relative to what you put out in your first decade of life. But in addition to this decline in numbers, there's a real qualitative decline in the thymus organ. So remember those MTECs, those medullary thymic epithelial cells that expressed all of the different proteins in our body, they really decline precipitously. So here we are looking within just the first 8 months. This is work from Ann Chidgey's lab. And we see similar things, that there's this very precipitous decline in these MTECs that express a lot of those diverse self-antigens within the first 8 months. And similarly, work from Ann Griffith and from Baron Gale has shown that if you look at the expression of those antigens, so they're comparing the expression of those diverse antigens. So if you see a ratio of 1, there's just normal expression, and we're looking at each dot as a different diverse self-antigen. If we look in the age versus the young thymus, you can see that many of those diverse self-antigens have declined in expression. And this is a more recent study that used some cool single-cell transcriptional profiling to see the same thing, that both, the self-antigens that would be expressed in your stomach or your eye or your pancreas, like insulin, right, are really declining. So if they're on the left side of the dotted line, they're declining with age. So that made us really wonder, okay, the cells that should express these diverse antigens and the antigens themselves are declining pretty early on in age. Is that aging thymus impaired in its ability to support tolerance? So very briefly, and I'm not going to get through all this, this is work from a former postdoc, Jessica Lancaster, a current postdoc, Damaris, and graduate students, Jayashri and you, and my lab manager, Hillary, in the lab. So we asked whether or not the thymocytes could get into the medulla, where they need to scan rapidly for all those diverse proteins. Is that impaired in the aged thymus? And does the aged thymus support efficient deletion of autoreactive T cells and Treg induction? Okay, so I'm going to move ahead and tell you that the way we do this is in mouse models in my lab. So we're able to take thymocytes of different ages from 1 month or middle-aged 12-month mice and load them onto live thymic slices that we've generated from 1 month or 12-month slices. So we can look at cells of different ages in different aged environments. And then we can take those cells to the 2-photon microscope to ask how well did the cells get into the medulla, how fast did they migrate? And when we do that, you can see kind of the beautiful migration of these maturing CD4 single positive cells. They're migrating within these medullary regions that are demarcated and labeled M, demarcated with the white line and labeled M. And you can see that both the 1 month and the 1 month in red and 12-month thymocytes are migrating within the medulla as they should be. And they're migrating pretty fast. These are 15-minute movies. So we've got this going on in our body all the time, right? The cells are really migrating around. And on the right are actually the same movies, just with the cells tracked. So the lines show the path of the cell. And so we asked, well, do the cells get into the medulla where they see all these diverse antigens as efficiently? And at first glance, the answer was no, because if we look in an aged thymus environment on the right, it looked like there was less entry into the medulla than in the young environment. But if we sort of zoomed in and looked at that, that actually had to do with this increased density of cells in the cortex rather than a decreased density in the medulla. And if you just look over here at this movie on the left, hopefully, there you go, there's my pointer. If you look at this movie on the left, you can see that if you look at the tracks, there's plenty of migration of cells in the medulla. So 12-month thymus, no problem. The maturing cells can get into the medulla, but they don't migrate as fast. And it doesn't matter how old the thymocyte is, how old that developing T cell is, the aged environment does not support rapid motility. Remember, they have a very limited time to find all those self-antigens. So is that enough to make a difference? Does it matter? Is that impairing tolerance in the thymus? So we went back to our thymus slice assays, again, different aged thymuses. And we took thymocytes that should undergo tolerance. And we looked at different types of tolerance to antigens that should be expressed by all the cell types in the thymus, or tolerance to antigens that should only be expressed by those MTECs. And after waiting, we can use flow cytometry, and I will skip what these flow cytometry plots are, but let's just say that that allows us to quantify negative selection and regulatory T cell induction. And what we can see here, what you're looking at is different levels of these highly expressed self-antigens. And in a 1-month thymus, as you increase the level of self-antigens, you get this decline in T cells because they're being deleted. So these are T cells that respond to this particular self-antigen. And in the 12-month middle-aged thymus, good news. By and large, if you've got lots of these self-antigens present, you're still going to get deleted. So the 12-month thymus can support some central tolerance and deletion of autoreactive cells. But when you start getting down to lower levels of the self-antigen, now the 12-month thymus isn't doing as good of a job as the 1-month thymus in getting rid of those cells. And as we look at these self-proteins that don't bind T cell receptors as strongly, what we start to see is a more profound defect. So now as we get to sort of these moderate levels of these broadly expressed self-antigens, the middle-aged thymus, which is still pretty robust, it's still exporting a decent number of T cells, is not starting to get rid of these cells. And a matter of fact, not only is it not getting rid of these cells, but there are more of them that are developing in the 12-month thymus. So you're taking some weak autoreactive T cells and allowing them to really expand in this thymus environment. If we look at the antigens that are expressed by medullary thymic epithelial cells, similar pattern emerges. So if we have a really strong medullary thymic epithelial cell antigen, no problem. The 12-month thymus in red does as good a job as the black 1-month thymus in deletion. But as we go to these weaker self-antigens, now we're seeing a real deficit in the ability of the red 12-month thymus to get rid of the autoreactive thymocytes. Okay, so that was all CD8 killer T cells. What about helper T cells? The same pattern emerges, except it's even more severe. So now, even with a ubiquitous strong antigen, the 12-month thymus is not good at inducing deletion, much less to these weaker antigens. And very notably, what you can really see down here in red is a real deficit in inducing regulatory T cells to these self-antigens. So now we're not as good at getting rid of the autoreactive cells, and we're not making regulatory T cells to suppress them in a middle-aged thymus to these moderate affinity self-antigens. Now note, if you're talking about a strong affinity self-antigen, that 12-month middle-aged thymus is not so bad at inducing Tregs to these strong self-antigens. It does an okay job. So it's not like it's completely impaired in its ability to induce regulatory T cells, but it doesn't do it to weak self-antigens. So why does that matter? Previous studies have shown, this is from Ed Palmer's lab, that these weak self-antigens that are expressed in the thymus don't do a good job of deleting thymocytes, as you actually just saw recapitulated in our own data, right? The strong self-antigens, you could see this real decline. I'll show you. Here's a strong self-antigen, great decline in the number of autoreactive T cells. The weak self-antigens, even in a 1-month thymus, you really don't get the deletion isn't as good. That's not shocking because the TCR isn't receiving as strong of a signal. But you do a really good job with those in making regulatory T cells to suppress those escaped autoreactive cells in a 1-month thymus. But you no longer do that in a 12-month thymus. And those weakly deleting antigens here are really good at getting out into the periphery and causing autoimmunity. So now in a 12-month middle-aged thymus, you're not getting rid of these weakly autoreactive cells, and you're not inducing Tregs to them anymore either. And if you look in people at middle age, this is where we really start to see in a lot of autoimmune disorders, as the thymus is starting to involute. Remember, I showed you that 28-year-old thymus, but there's still tissue there to pump out a good number of T cells. This is where we start to see an increase in incidence of autoimmunity in people, whether in this case, we're looking at lupus or here at multiple sclerosis. So these are often diseases that arise in sort of young middle age in people. So now what we're really curious about is, all right, I'm going to skip this for the interest. Well, okay, we want to know what's going on here, right? So why is this middle-aged thymus becoming less capable of supporting tolerance of our T cell pool? And we're going to use a variety of tools here to look at whether or not interactions with the different dendritic cells and these medullary thymic epithelial cells are impaired in the middle-aged thymus. We're using the pretty rainbow in the middle is a really awesome new technique where we can actually look at all of the genes expressed by individual cells. So every dot here is a cell, and we can really zoom in and ask about transcriptional changes and cell subset changes on the total transcriptome level to understand how these antigen presenting cells that get rid of or tolerize our T cells are changing with age. And then we're using sort of kind of newer multiplex immunofluorescence to look at all of these different subsets and how they're spatially localized with one another and how they're interacting and how that changes with age. So that's it. I want to stop and acknowledge my wonderful lab members without whom none of this is possible. And I think I already mentioned Jay, our grad student who worked on the project I was talking about, Damaris, the first author on our paper about diminished central tolerance that I think just came out yesterday. So I wanted to tell you a little bit about it because I'm excited about it. Sahi, a postdoc who's contributed to these studies and also does a lot of our ALL work. And Hillary, where is she? Yes, hiding in the back, but definitely not least at all is my lab manager, Hillary, without whom none of my lab would run, and who also contributes to all of our multiplex imaging. And I want to thank my collaborators at multiple institutions. So Ellen Ritchie, Nancy Manley, Esther at UT, Esther Malamed for the COVID-19 research, Pete Savage at the University of Chicago, Matt Bettini, Janko Nikolic-Zugic, who's been really key in aging our aging T cell story, Marcel Vandenbrink at Memorial Sloan Kettering, and Jared Dudikoff at Hutch, and Laura Hale and Greg Simpowski at Duke, as well as collaborators on other projects. And I want to thank my funding sources, so the NIA and NIAID, as well as our cancer funding sources, though we didn't really talk about that. Okay, and that's it. We're almost out of time, but I'll answer anything that I have time for. There we go. Thank you guys for listening, and hopefully you found that interesting.
It was very interesting. Thank you so much, Dr. Ehrlich, for joining us today. I think we have literally one question, so it's going to be rapid fire, very, very short. Is it possible that increased autoimmunity in middle age may actually be helpful at targeting emergency cancer precursors? So you've kind of talked about all these different things. So how does that?
That's such a great question. And there is evidence. So particularly like in bone marrow transplants with leukemia, there was this real observation that if you don't deplete the T cells during a bone marrow transplant, then they're going to cause graft versus host disease and attack the host. And that's really detrimental and bad. But the flip side is that in leukemia patients who received a bone marrow transplant, one thing that was noticed is that if you left a little bit of the alloreactive, the T cells that could respond to the recipient of the bone marrow transplant, that actually helped to fight off the leukemia itself. So it's a little bit of a different point, but it's highly related, which is that maybe some of that autoreactivity could actually help protect against cancer. And I don't know. I don't think that's been explored, but I think it's a really great idea and possible thinking about those experiments. Yeah.
Okay, that was our one question because we're right at time. We did have a lot of great questions. We'll try to capture those so you can take a look at them. We'll do our best to do that. Thank you to everyone for joining us today. We really appreciate you for taking the time to join us with Texas X's, and it's always a pleasure to have our lecturers and our attendees. And just a reminder, our June lunchtime lecture is in the works. So stay tuned for more information on our website at texasx's.org/lunchtime lectures, and we'll get that information to you ASAP. Again, thanks for joining us today, and everyone have a great week.